Hi everyone, my name is Taylor Shirley and this is my presentation on high-content microscopy. High-content microscopy is a form of fluorescent microscopy that looks at the dynamics within a cell. This high-throughput method allows researchers to study protein-protein interactions, biochemical processes, and more within a singular cell using fluorescent tags to visualize the various targets. The controls used in a high-content microscopy experiment are an autofluorescent control, which does not contain stains or antibodies and is otherwise treated the same way as samples, making any fluorescence a background signal, and a secondary-only control for all secondary antibodies used, which is used to test for any non-specific binding within the reaction. An alternative to high-content microscopy is light sheet microscopy. Some advantages to high-content microscopy are that it has high resolution, allowing for clearer images, and that it has the capability of rejecting out-of-focus light so that it will not contribute to the overall image. Some cons, on the other hand, are that it only works with single-cell samples, and it has a low sensitivity, and that it is too harsh to work with living cells. Some advantages to light sheet microscopy are that it works with larger samples, it has a high sensitivity, and that it is gentle enough to work with living cells. Some cons to the light sheet method are that it has a low resolution and that it produces large data sets due to the large samples that can be difficult to analyze. Here we have an image from a paper titled Automated Microscopy for High-Content RNAi Screening. In this image, figure A shows the detection of cells based on fluorescent chromatin labels indicated by the red areas. B shows manual annotation of morphology classes with the interphase nuclei being annotated by green asterisks and the metaphase chromosome plates annotated by the yellow asterisks. C shows the hyperplane that separates the two morphology classes, which is automatically determined by a classification algorithm. D shows that the classifier can be used with new data for automated classification using the same annotation key as figure B. E shows a time result assay for mitotic progression with annotation being done by machine learning. On this slide, you see an image from a paper titled Microscopy-Based High-Content Screening. In the figure shown, A shows the scanning electron micrographs of adult Drosophila eyes with the wild type on the left and a transgene introduction causing the rough eye shown on the right. Figure B shows the images of cells after staining, where blue indicates DNA and green indicates A-tubulin, showing the effects of monostrol treatment on spindle formulation. C shows cell stain for DNA, A-tubulin, and actin to visualize phenotypic features. D shows Drosophila eyes stained as shown in figure C for analysis of morphological phenotypes. Figure E shows the confocal micrographs of cells infected with influenza virus and stained green for the virus, stained red for CD63, and co-localized virus particles and CD63 stained yellow. F shows confocal micrographs of L cells after treatment with WNT3A conditioned medium, where magenta indicates DAPI and green indicates lipid droplet formulation. Here we have an image from a paper titled The Potential of High-Content, High-Throughput Microscopy in Drug Discovery. The figure shown indicates the detection of multiple fluorophores in one cell, showing the nucleus, cytoplasm, microtubules, Golgi complex and transport carriers, and the endoplasmic reticulum individually, and then combined for the final image to show the fluorophores present in each of these areas. Thank you for tuning into my presentation. I hope you enjoyed it, and if you have any questions, please do not come to me. I do not have answers for you.